Good afternoon and welcome to Well Aware with Michigan Medicine, a series of community discussions to keep you informed. I am Kelly Malcolm, lead research communicator for Michigan Medicine's basic science departments. Thank you all for joining us today. We sincerely want to thank all of you for the many ways you provide support to Michigan Medicine. Let me also say thanks to those who submitted a question on the RSVP form. We will be working to answer many of those questions later. We will not, however, be able to collect questions live during today's webinar. Ending the COVID-19 pandemic requires understanding as much as we can about the virus behind the disease, as well as the science behind its various effects on the body. Michigan Medicine's researchers are hard at work investigating fundamental questions about this novel coronavirus, SARS-CoV-2. At the same time, researchers are using this knowledge to inform the development of new treatments, to discover existing drugs that might be repurposed to fight this novel virus, and working to produce vaccines to stop the pandemic and prevent infection. In addition, U of M is committed to tackling other infectious disease threats, both current and emerging. Today, you will hear more about these efforts from our experts. Now to introduce our panel. We have Bethany Moore, Professor of Microbiology and Immunology and Internal Medicine, who will touch on the contributions of basic scientists to develop a better understanding of the biology and treatments for SARS-CoV-2 and other infectious diseases. Johnny Sexton, Assistant Professor of Internal Medicine in the Division of Gastroenterology and Hepatology, and the Director of the U of M Center for Drug Repurposing, who will present on drug repurposing for COVID-19. John Trainer, the Edward F. Domino Research Professor and Professor of Pharmacology in the U of M Medical School and Professor of Medicinal Chemistry in the College of Pharmacy, who will discuss studies using a novel virtual screening method to identify drug targets in the SARS-CoV-2 virus and to examine FDA-approved drugs for interactions with these targets as potential treatments for COVID-19. And Adam Loring, Associate Professor in the Division of Infectious Diseases and the Department of Microbiology and Immunology, discussing the basic science behind COVID-19 vaccines. Now, I'd like to invite Dr. Moore to join us for her presentation.
Thank you very much. It's my pleasure to be here. Great. Well, as I said, it's really my pleasure to be here today and to highlight for you of the way basic scientists here at the University of Michigan are engaged in the fight against COVID-19. If I could have the next slide. Great. First, I just think I want to define what I mean when I say basic science. And really what basic science is any investigation that seeks to understand, to increase knowledge or understanding of a disease process or a biological process. And often that work is really focused on trying to understand causal mechanisms of health and disease. And it involves scientists doing experiments to test our hypotheses, or what you can think of as our theories about how the system might be working, particularly in the case of this virus. And in the medical field, these experiments often require the use of model systems that can be very easily manipulated. And some of those are shown on this slide and include things like yeast, fruit flies, zebrafish, and often for the study of pathogens, mice. And I think it's important to highlight that the results from such studies can really help to inform future clinical studies as well. Next slide, please. So you might be wondering why mice are useful for the studies of COVID-19. Well, you may not know, but actually mice and humans share about 85% homology in their genomes. And also the physiology of mice and humans is actually quite similar in the way that the organ systems work, and importantly in the way the immune response responds to pathogens. And the other thing is that we have used mice for a very long time, and so we have wonderful experimental tools that we can really use that are well characterized that can, for example, use an antibody that can help us identify what cell types are involved or what kind of molecules are being secreted by a particular cell type. And because we've used mice for so many years for so many different studies of pathogens, we can learn a lot from those studies to help make inferences for what we're doing right now. Certainly one of the greatest discoveries in my time as a scientist has been the ability to genetically manipulate mice. And that gives us the power to be able to change a single gene and be able to study how that changes the response to a particular pathogen, like a virus, for instance. And really, some of those studies would just never be possible in humans. And one of the things that makes studying a disease like COVID-19 so challenging is that the disease symptoms are caused not only by the ability of the virus to replicate and cause damage, but also by the host immune response, which can also sometimes cause damage. You'll see on this slide a very simple schematic that just shows in the body and how that cell can then help to recruit and activate the adaptive immune cells. You'll see here in green the cytotoxic T cell, and in blue a B cell. And what you can see diagrammed here is under the B cell, you can see these antibodies that are being formed to the virus. And if they're effective in neutralizing, they might be able to prevent the virus from binding to other cells and causing additional infection in the host. Whereas if the host cells are already infected by the virus, you'll see that the green cytotoxic T cells are now the kind of cell type that we might need to actually be able to clear that. So we can use these kinds of model systems to really try to understand what features correlate with protection from disease versus worse disease. And that really helps us understand the human situation as well. Next slide, please. Now, as powerful as these model systems are, there are definitely challenges. And in particular, this particular virus, SARS-CoV-2, gives us a lot of challenges. So you may know that the virus itself binds to the human ACE2 receptor, which is shown in this diagram here. You can see the ACE2 receptor in green. And that's like a lock and key mechanism, because once the virus binds to that receptor, that allows the virus to then gain entry into the cells, into many different tissues within the body. And despite the fact that humans and mice have about 85% homology, this is one situation where this SARS-CoV-2 virus actually does not bind to the murine receptor. And so that makes it difficult to study because it won't infect mice that we haven't done anything to. The other thing is that this virus, because it causes such severe disease in some patients, has to be handled under what we call biosafety level 3 conditions, which means that those are in a negative pressure room, self-contained with all the equipment that stays in that room. And then you have to use special equipment and a respirator so that the researcher is protected from working with the virus as well. Next slide, please. All right, so despite, as I mentioned, there are some challenges. So how are we overcoming those challenges? Well, as I mentioned, one way is to be able to genetically engineer the human ACE2 receptor into mice with accurate tissue expression so that we can allow the virus to infect mice and then study those responses. And in fact, there's a colleague of mine here in the Department of Microbiology and Immunology, Dr. Phil King, who is actually working on that right now. Another thing that we really need to do is to build a BSL-3 facility that's large enough to house animals. And while we are really actively raising money to do that here at the University of Michigan right now, we don't have it yet. And so until then, we're collaborating with other investigators at other institutions where those facilities are available. A final approach is what my lab is doing, and we're studying a genetically similar coronavirus that can infect mice and in fact causes a very COVID-19-like disease. And we think we can make inferences about the responses we see there to be able to tell us something about the disease in humans as well. Next slide. So I've mentioned a lot about mouse models, but that's certainly not the only systems that we're using. We can collect lung, intestine, or kidney tissue, for instance, that are discarded after, say, surgical procedures. And we can use those tissues to build these small organoids, as you'll see on this slide. This is a lung organoid. And these are just small sort of clusters of three-dimensional tissue architecture of cells, seen here making up of both epithelial cells as well as fibroblasts. And we can use these cell types, or these organoids, to infect with the virus and then study how the virus replicates and how the tissues respond. It's also possible to use cells that we collect from humans and can keep alive outside the body to study the mechanics of the viral replication in as well. And these approaches have been very important for us to try to understand how the virus enters cells, how it replicates, and certainly it's important for tissue-specific responses. And I think as you'll hear later, they're also very important for allowing testing of therapeutics. Next slide. So now that I've told you a little bit about the approaches we're taking, I just wanted to mention some of the questions that we're trying to tackle here at Michigan. We're trying to understand how the virus is mutating. We want to understand why the viral infection is worse in patients with diabetes. We want to understand how the virus causes the vascular complications and the thromboembolic events that you've probably heard about. We're trying to define what vaccine elements would make the very best vaccines across a very diverse population with different kinds of genetic backgrounds. We're trying to understand what surface coatings might be best at resisting the virus so that they wouldn't pass transmission as easily if you were to touch that surface later. And certainly we're trying to understand how we can detect SARS-CoV-2 much more quickly and accurately, and that's just a few of those questions. Next slide. So we've talked a lot about SARS-CoV-2, but I just wanted to ask the question, if we could start this poll, what other infectious diseases worry you? I'm just curious to know whether you think about influenza or maybe avian influenza, HIV, Ebola. What about antibiotic-resistant bacteria, sometimes called the flesh-eating bacteria? What about tick-borne viruses? Things like West Nile, or as we've often heard in Michigan just recently about the Eastern equine encephalitis virus. Maybe malaria or all of the above? Can we see the results to that poll? Well, it looks like we have a lot of concerns out there. And maybe the most important thing I can tell you is that those same diseases worry us as well. So if I could have my last slide here. Researchers here at the University of Michigan are studying many kinds of viruses, many different bacteria, parasites, and fungi as well. And what we're really trying to do is to understand how those pathogens cause disease so we can learn how to treat and cure them as well. And I'm sorry, I misspoke. My very last slide is the next one, which really is just to remind you that we have deep expertise in many areas related to infectious disease here at Michigan. So we are working hard to do our part to stop this pandemic and to prevent the next one. And with that, I'll turn it over to my colleagues.
Thank you so much, Dr. Moore. You provided a really helpful look at how researchers are learning about the fundamentals about this virus. And now I'd like to welcome Dr. Sexton to the stage for his presentation.
Hi, Kelly. It's really great to be here with you all. Can everybody hear me? Great. So I'm really excited to tell you about an effort that we started back in early March when it was really apparent that the COVID-19 pandemic was not anywhere close to being over. And the question that we were asking was that, can we repurpose existing drugs to help combat the COVID-19 pandemic? So next slide, please. So this is the translational research cycle. So Dr. Moore gave a great introduction to basic science research, and that's what we call the T0 phase here, preclinical research. And this is absolutely essential, and it lays the groundwork for the steps that have to come next, which is translation to human beings. So this is in early stage clinical trials where we make the hypotheses from basic science research and then translate those to the clinic. And then the later stages in clinical translation basically are just going to larger and larger clinical trials and then ultimately translation to the population at large. And so, but the biggest barrier is between this T0 and T1 phase here. So one of the approaches that can help to just absolutely jump over that hurdle is to repurpose existing drugs where we have human safety data. We know these drugs are out there and that are safe. So discovering new uses for these drugs can be very rapidly implemented in the clinic to, to respond to something like a pandemic. So we founded the U of M Center for Drug Repurposing in late 2019, and then we're presented with this absolute moonshot for drug repurposing. And that was to find existing drugs that are commonly prescribed that may have efficacy for COVID-19. And when I say that, what I mean is that if we can find drugs out there that have either a direct antiviral effect or that can help suppress the inflammatory response, that can help keep people out of acute respiratory distress or take them from being symptomatic to asymptomatic. And so that's exactly what we tried to do right at the beginning of the year. Next slide, please. So how do we do this? Well, as Dr. Moore said, we needed a model system. In that model system, the best model system you can have is a human cell that gets infected with live SARS-CoV-2 virus. And she mentioned that this work needs to be done in a field of human cells. These are live human cells that have been infected with the SARS-CoV-2 virus. So what we did was in early March, we decided that this is something we were going to go after. In early April, we obtained the virus. So this is the WA1 strain. So this was patient 0 from Washington. And that was the first American who had contracted SARS-CoV-2. And so we were able to obtain this virus and then actually infect human cells with this. And when you look at this picture, it may be obvious to you which are the infected cells. And so what you see in cyan here are all of the nuclei of cells. And then those cells that look purple or magenta, those are the infected cells. And so this establishes a model system that we can use to find therapeutics that can reduce this infection. So in an ideal experiment, what we would see is we would see that these purple infected cells would just disappear. They would not happen in the first place. So we sought out to do this. The first thing we needed to do was to collect all 1,425 FDA-approved drugs that are commonly prescribed. So we did that at the end of 2019, and so we were perfectly poised to conduct this study. The next thing we did was to develop a human cell infection model, and that's the result of that you're seeing here in this image. But because there are so many drugs and we have to take so many images to detect drugs that have some antiviral effect, we had to use artificial intelligence, specifically machine vision, to go and look at these images for us and tell us when they see this reduction in infected cells. And then, of course, the final step is with any in vitro result is that we have to translate that into human beings in a safe and an effective manner, which means that we need to do randomized control trials, clinical trials, to establish that these are safe and effective in human beings. And that's really where we're at right now. Next slide, please. So this is another image of SARS-CoV-2 infected human cells. And what I see here is just an amazing array of different kinds of cells. So what you see is you see very small round cells, and then you see other cells that have these really wild noodly appendages. Now, these are human cells. They normally don't have these kind of appendages. This is something that the virus is causing them to do. And what we see is that we see some cells get a direct infection from a live virus, and then other cells get the infection by the cell-to-cell spread. And this imaging technology allows us to see all of that detail. And as they say, a picture is worth 1,000 words, but this picture is worth about 10,000 data points. And it's this coupled with artificial intelligence that helps us make good decisions about where compounds are working and what their mechanism of action might be. Next slide, please. So just to introduce you to this assay technology. So in the laboratory, we call a test, we call it an assay. And normally, these can be done in Petri dishes or test tubes, but in this particular experiment, we use what are called 384 well plates. So I've got a 384 well plate right here. And I don't know if you can see this, but there are 384 individual places where we can do experiments. So what that means is that we can test that many drugs in one single plate, and then we can stack these plates up. So this is a fully automated process that uses robotically controlled microscopes to capture these absolutely beautiful images that you see. And so we use fluorescent dyes to detect the virus. We did 24,000 tests in 6 weeks, collected 1.5 million images, and then used AI and machine vision to detect infected cells. So in every good experiment, we have controls. So we know what uninfected controls look like and infected controls look like. So this is one of these 384 well plates here. And you can see that in each one of these wells, we take images like this. And this is the machine vision part where we identify every single nucleus and then also identify the infected cell population. And this is exactly how we detect efficacy, by looking to see for a reduction in that population of infected cells. Next, please. Just to give you an example of that, this is remdesivir. This is a drug that everyone's heard about, that has been used successfully in certain contexts. And so you can see this is an infected cell population. You see those purple guys. And then in a remdesivir treated well, you can see that that virus just completely goes away. So this is what efficacy looks like to us. Next, please. So I want to tell you about a couple of drugs that we're very excited about. We discovered many with antiviral efficacy, but there are a few that I want to tell you about just very quickly. So lactoferrin, it's a widely available dietary supplement. So this is available at health food stores. And this is a natural antiviral system. So lactoferrin is a protein that's found in colostrum breast milk. And this was actually just recently advanced into clinical trials and showed some preliminary signs of efficacy. So this is really good. What this means is this can be a very safe and effective treatment. And we're advancing this one into clinical trials as we speak, into a larger trial, so that we may establish that we have efficacy in a large human population. There's other drugs called fluvoxamine, clofazimine, and Atrovent that we're also very excited about advancing into clinical trials. Atrovent it's an asthma rescue drug. It's found in inhalers. Clofazimine is actually used for Hansen's disease or a leprosy drug. It's also been demonstrated to have efficacy. And fluvoxamine is an antidepressant. So next slide, please. So the next steps are really just what I said. We're advancing these laboratory results into clinical trials, and if you stay tuned, we'll have results hopefully in just a couple of months that will be able to provide small molecule drugs to the community to help combat this pandemic. So with that, thank you very much.
And thank you, Dr. Sexton. You presented some really exciting prospects for possible treatments for helping those with COVID-19. And now I'd like to welcome our next presenter, Dr. Trainer.
Hello, everybody. As we've heard on the news and from Dr. Sexton, many ideas are forthcoming about ways to treat COVID-19. Indeed, many of you will know that Donald Trump, for example, has been given an antibody cocktail, taken hydroxychloroquine, remdesivir, and vitamin D. However, several of these are not proven treatments or easy to administer outside the hospital, so it's absolutely essential that our search continues, not only for effective vaccines, but as Dr. Sexton just mentioned, also for drugs to treat infected patients, as well as the identification of targets in the virus that may be susceptible to drug treatment. Finding new drugs incorporates traditional methods of chemists designing molecules to act with disease targets, of experimental screening of thousands of compounds against disease targets, for example, proteins in SARS-CoV-2, and virtual screening methods where supercomputers are employed to examine literally millions of often theoretical compounds against a specific drug target. When drugs are identified, these virtual methods, they are only usually very weakly active, but can be chemically modified to improve potency. These techniques have been applied to COVID-19 by several groups and have led to the recognition of potential new candidates. But today I want to illustrate how two postdocs in my lab, Keith Olson and Wallace Chan, are using a variation of this technique to start to identify possible mechanisms against SARS-CoV-2. However, as you're probably aware, new drugs, and as Dr. Sexton mentioned, new drugs can take many years of clinical and especially safety trials before they can be widely available to the general public. Thus, like Dr. Sexton, we decided to investigate only drugs that will be readily and quickly available. That is drugs that were already approved by the FDA, the Federal Drug Administration, and which many of you may already be taking for other conditions. However, this approach by myself and Dr. Sexton does mean taking drugs for a purpose for which they were not originally intended, along with any side effects they may have. So I just wanted to gauge how people felt about this. Could I have the poll, please? Okay. Do we get a number yet? I have to wait a second for everybody to vote, I think. Wow, it's taking a long time. 73% have said they would feel comfortable. Only 2% said maybe. So, 2% said no. So I think that's pretty good. I mean, if everybody had said no, we would rather be wasting our time perhaps, but that's a good thing. So the next question that arose once we decided to do this was, how do we target the virus? Which particular component of the virus makeup would be the most susceptible? Dr. Sexton showed you results where his group looked for changes in the images of cells in the presence of the virus. However, for a computational search, we needed a specific target. We also did not believe any of the originally approved drugs, the already approved drugs for any disease condition, would be magic bullets. That is, we did not expect them to kill the virus stone dead. If so, I think these would have been already identified. We reasoned we would find drugs that have had a better chance if we identified a cocktail of 2 or 3 molecules that might alone have weak activity, but either A, attack the same protein in different ways, thereby giving a better chance of deactivating this protein and its role in viral replication, or B, attack several different proteins in the viral life cycle. Thus, by combining drugs, we felt it might be possible to get a synergy where the sum of activities was much better than the sum of the individual drugs. And it also, under these circumstances, it might be more difficult for the virus to develop resistance. In my lab, we're used to investigating proteins, so we decided to virtually examine as many of the 29 proteins making up the virus as possible. Proteins, however, are very large molecules compared to the drugs, which are usually very small. Next slide, please. Norris, so back one, gone too fast. So Keith identified potential places on each of the proteins where we might target drugs. Many scientists are working to identify the structures of the SARS-CoV-2 proteins and making these available. And many of the proteins are enzymes with active sites that can be identified. In addition, Keith searched for sites that might potentially bind small molecule drugs. Little pockets or clefts in the protein are shown in this slide. And again, this is just a snapshot of the protein. It's probably 5 times this big, and you can see how small the drug is, but it does fit in that nice little pocket there. Pockets or clefts that could possibly alter the shape of the protein and interfere with its action. This led us to identify 48 sites on the SARS-CoV-2 proteins, and obviously for that reason, so had several sites. Next slide, please. Wallace then took these sites and determined how well FDA-approved drugs bound to the proteins, illustrated here for one protein. In this example drawing, the bottom 3 drugs fit very well into a pocket on the proteins, but the top 5 do not. This exercise was repeated against the other 47 protein targets. On the face of it, this sounds like a fairly trivial exercise, but actually required the use of the Great Lakes supercomputing facility, and it would have taken 24 months of computing time if we had done the experiment sequentially. So it was actually a huge amount of work. But the result of this was the identification of 246 drugs that could potentially bind to viral proteins. We then compared these drugs with drugs identified by Dr. Sexton's drug repurposing study. This revealed 18 compounds that showed up in both systems. Changes in the cell. So this could include protective effects on the host cell, effects on the systems in which the systems in the cell that the virus needs, or effects on the cell itself, whereas we have only looked at the viral processes. Also, of course, there's a difference between the reality and the virtual reality. And of course, doing large computer studies like this, you have to make certain assumptions in the process. But one way we can provide further supporting evidence for our virtual studies is to ask how stable the interaction between the drug and a particular protein. Proteins are not static molecules, but continually change shape as chemical bonds holding the protein together stretch, break, and reform. With the help from Dr. Heather Carlson in medicinal chemistry, we studied these movements and the stability of the drug-protein interface using a technique called molecular dynamics, which simulates the natural movements of the proteins. Could I have the next slide, please? If you look at this picture on the left, you can see that this drug with this cleft in this protein is very stable. As the protein moves around, it stays there. But in contrast, the one on the right, as soon as the proteins start to move, it flies off. So using this technique and picking the compounds that will seem to stay longer in the binding pockets, we reduced our virtual drug candidates down to numbers to 6. Next slide, please. This slide illustrates a so-called heat map of the interaction between the target proteins, which are listed up the side of this figure, to the 6 drugs that we identified listed on the bottom. The red bars represent what the computer tells us is a strong interaction, and the blue bars, the dark blue bars, indicate no interaction. But you can see we do have a selection of compounds that hit one protein in different places. For example, drugs 3 and 5 bind to the spike protein and different parts of the spike protein even, which is responsible for the entry of the virus in the cell. In contrast, compounds 5 and 13 bind to different proteins. So on the final slide, please, we see that all of our studies are theoretical, relying on data generated by computer hardware and software, albeit based on basic concepts of physics and chemistry. Our next steps are actually to test the ability of the compounds to bind to the predicted proteins, alongside testing combinations of the drug, which we predict is what we'll need, against the virus in human cells, as described by Dr. Sexton, and organoids, as described by Dr. Moore, and hopefully we will find active combinations. I have not mentioned any specific drug names. This is because so far this is a purely theoretical study to illustrate what can be done, and we have not yet concrete evidence against any of these compounds or combinations. But it was intriguing to us to find that one of our 6 compounds is a vitamin D supplement. And as you probably know, vitamin D does have general role of supporting the immune system. And a recent study has indeed linked vitamin D deficiency with susceptibility to COVID-19. Thank you very much.
And thank you, Dr. Trainer. It's very encouraging that there may be existing drugs that could help us against this new virus. And finally, I'd like to welcome Dr. Loring.
Great, thank you. It's great to be here. Next slide, please. So my job today is to tell you about the basic science behind vaccines. You heard from Dr. Moore a little bit about how we study immunity to viruses in mice, and basically, we try to use that to figure out how to design the best vaccine. And so the first question we ask is, how does the immune system protect you from viruses? Usually, as soon after someone becomes infected with the virus, the immune system recognizes the invader and kicks in. And cells will secrete these molecules called interferons, which signal to other cells that there's something wrong here and they need to engage in the fight against these invaders. And these interferons and what happens later due to interferons are what make you feel sick, what cause your fever, maybe aches, things like that. That's actually your body recognizing and responding to the virus. Over the next week or so, we start having what's called an adaptive immune response. And these are two different flavors of immune responses where you have these cells called B cells, which make antibodies. And these antibodies sometimes coat the virus and then protect you from the virus infecting more cells. There's also immune cells called T cells, which are very good at recognizing other cells in your body that are infected with viruses. And then the T cells come in and kill those cells. And so by killing the cells, you essentially help get rid of the virus. And so those are just a few ways that our bodies respond and hopefully are able to clear these viruses so they don't cause more damage. Next slide. So the main idea of the vaccine is to trigger these same responses without having to go through the trouble of being infected. And so the gold standard is to see if it actually prevents people from getting infected or getting sick. And that's what the big clinical trials are doing right now. They're trying to figure out, short of that, you want to figure out what immune responses are triggered by an infection, and then how to reproduce those same immune responses. So what parts of the virus trigger an immune response? What immune responses seem to prevent infection? And when is an immune response too much, such that it causes damage? And this builds on decades of work and understanding how our immune systems respond to viruses. So that gave us a big head start in figuring out how best to protect against COVID. So what I've just schematized here is graphs that you will see in many of the papers describing these vaccines that you've been hearing about in the clinical trials. And so people will measure an immune response, whether it be interferons, those molecules I told you about, T cells or antibodies, and compare them between people who are infected and recovered. And so that's what your natural infection triggers. People who didn't get the vaccine, the placebo, or people who vaccinated. And so you look to see what kind of vaccines best trigger immune responses that best match ones from people who are infected with the virus. So next slide. So there's been a lot of talk about Operation Warp Speed, and it's truly amazing how quickly we have vaccines in these late-stage clinical trials. It's also remarkable that the vaccines that are in the latest stages of development take very different approaches for most of the vaccines we commonly receive, such as the measles vaccine or the annual flu shot. And that they build on years of research into how to safely deliver parts of a virus in a vaccine and how to focus the immune response on that piece of the virus. In the case of SARS-CoV-2, most of the vaccines are based on the spike protein, which is on the surface of the virus. And so I've shown here some of the new vaccine approaches which derive from years and years of research. So in one, you start with another virus, so an adenovirus or a virus called VSV, and these are called vector. And what you do is you take these viruses and then you take out key genes that the virus needs to copy itself. And that way that virus is much safer. It doesn't cause any disease because it can't reproduce. And then you take that and you put in the spike gene from SARS-CoV-2. And then you have you're essentially using these other viruses as delivery systems to deliver the gene for the spike protein into your cells in your body. And then your body can respond. Another way is what you might have heard of from the Moderna vaccine or the Pfizer vaccine, which these are RNA vaccines. And RNA is the message that tells a cell how to make the spike protein. And these vaccines are essentially the RNA that are put into what's called a lipid envelope. It's basically just a little fat droplet and millions of little fat droplets. And then those get delivered into cells. And this is the technology is based on years of research in how to RNA delivered to cells and how to get that message in. Finally, there's another one from Novavax, which is a protein subunit vaccine, where they actually just take the protein from the virus and package it into these nanoparticles, which help stimulate a stronger immune response per unit protein. Next slide. So most vaccines rely on the spike protein. Last time I spoke to you, I talked about how we were using sequencing of viruses to track how it's mutating over time. Next slide. And so what people do now is they look at those mutations, and here's one in the spike protein, and they see, does that change how the immune system responds? And so people will recover viruses from people who have different mutations in the spike protein, and then test them and see if the antibodies still work. And that's important for seeing whether we need to change what's in the vaccine or not from year to year. The good news is it looks like everything is working just fine, and these mutations don't seem to change things too much. Next slide. So I hope I've been able to show you how the rapid developments in COVID-19 research here and elsewhere actually build on years of work on basic virology, immunology, and molecular biology. Without that foundation, we wouldn't be in the position we are today with a vaccine likely available to some extent within the next 6 to 12 months. Recognizing the importance of fundamental science and the approaches needed to combat emerging diseases like COVID-19, the University of Michigan is establishing a new Michigan Center for Infectious Disease Threats. We have existing strengths across the campus in this area, if you've seen. So in Michigan medicine, in terms of virology, immunology, infectious diseases, in the School of Public Health and Infectious Disease Epidemiology, Modeling, and Vaccine Research, at the School of Pharmacy with drug discovery and formulation, and LS&A with disease ecology, chemical biology, and molecular biology, and the College of Engineering with strengths in nanomaterials, advanced diagnostics, and aerosol biology. And of course, there's much, much more at other units across campus. And so the goal is to build bridges throughout the University of Michigan campus to tackle these important problems with emerging infectious diseases. Thank you.
And thank you, Dr. Loring. Just by their very nature, infectious diseases are of concern to all of us, and it's nice to know that U of M is leading the way to understanding and defending against them. And now it is time to bring all of the panelists back to the stage to address some of the questions and topics that have been submitted by our audience. All right, I think we have everyone. Okay, so into our first question. It's a rather broad one. In the early months of the pandemic, we heard a lot about how there was unprecedented cooperation among scientists worldwide to address the challenges presented to us by the virus. Does this continue? If not, what happened to change the cooperative attitude and behaviors? If it does continue, can we expect it to have an influence on the bigger world of scientific inquiry going forward? Dr. Moore, do you want to take that one?
I'd be happy to take that first. Well, I think it's true that there really has been an unprecedented amount of cooperation and speed with which scientific results have been shared with the community. Some of you may know that within, I think, 10 days of the time that the virus in China was reported to the World Health Organization, the virus had been sequenced and the sequence had been shared with the scientific community. And that was critical because it allowed other researchers to be able to start to do the bioinformatics analysis that you heard about today to try to predict vaccine candidates, to try to predict drugs that might work, things like that. And I think that that cooperation is still continuing in that one thing that's really, I've seen a lot of in my field is people starting to put their results on what are called these preprint servers like bioRxiv, where they put the results of their experiments out there very quickly before they've gone through peer review and publication. It's both a blessing and a curse because you have the access to data very quickly, but of course you have to then be very, very cautious about how you interpret all of those data because they haven't been through the normal peer review process. But I think that that willingness to share has been really critically important in helping us move as quickly as we have, and is really something I think that's been quite unique during this particular pandemic.
Great.
Can I just reiterate that? I think the research to develop the SARS-CoV-2 drugs relies on the sharing of information on the basic biology of the virus and its proteins. We'd be completely in the dark without it. People that were working in the old ways, just doing their own thing and keeping it to themselves, we would be nowhere. Should also add that the new methods of publication and availability of data before peer review, like Dr. Moore suggested, is allowing science to move much more quicker. And that's essential when you get faced with the challenges like the virus faces, or indeed, opioid pandemic or epidemic and things like that. In addition, I think the solving of scientific problems is no longer feasible by scientists working alone. So collaboration is essential, integration of ideas is essential, and experimental techniques that people can share is absolutely necessary. So I really hope this freedom of sharing information continues.
Great. On to our next question. Why does there seem to be so much debate among scientists concerning the transmission, treatment, and vaccine development for COVID-19? Dr. Loring, do you want to try and answer that?
Sure. I guess I can understand that this impression. However, I think it might be not actually the case, or that alarming, I guess. There'll always be different views on how to tackle an important scientific question or approach a complex problem. That's what science is often about. On top of that, this is an entirely new disease, so people are grappling with a lot of unknowns very quickly. We're only 9 months or 10 months in here. And then on top of that, there's a lot of media attention, so everything gets covered. So this is happening all the time, but we're just seeing it a lot because there's a lot of newspaper articles and news reports about these very questions. And so it can make things seem more chaotic, I think, than they really are. And I think there actually is, when you dig deeper, there's a lot of common ground in how scientists are thinking about a lot of these questions that you outlined, and emerging consensus in a lot of areas. But then there's still new things that people are grappling with, but eventually the science will kind of, again, get to a consensus on these later questions.
Can I just make a comment? I think I agree with, I definitely agree with Adam, and appreciate all the science that's being done to bring together an understanding and manage of this virus. I think the trouble is it took us, as Adam just said, by surprise. It's not very long ago that it was there. And so there was obviously appeared to be confusion as people developed hypotheses about the virus, tested those hypotheses. But I think as we move forward, and if we allow the science and medicine to direct us, we'll be along the right path. And I think it is coming together. It's just that, people looking at different aspects of it and trying to get a coherent pathway. And I think we're getting there.
I might just add that I think it's very rare to have so many scientists all across the world focused on a single question and a single problem. A lot of us work on our individual diseases or our individual questions. And really what's unprecedented about a pandemic like this is that everybody's bringing their expertise to bear on it. And I think that's where you get all of these different ideas and sharing all of these different theories and different hypotheses. It's really just because of so many people really tackling the same question at once, which is also, I think, why we've made progress as quickly as we have.
Great. Okay, so on to our next question, which is a rather practical one. Is it necessary to wipe and disinfect surfaces, groceries, and the like? How long does the virus remain viable on a surface? Dr. Moore, can you answer?
Yeah, I can tackle that. So it's a great question. And what we know is that under idealized laboratory conditions, that a virus can last on, say, a stainless steel or a plastic surface for maybe 2 to 3 days under those perfect conditions of humidity and things like that. Now, certainly if you're outside, air currents dissipate the virus more quickly. UV light from the sun can help to eradicate the virus more quickly. And so I think it really is hard to know exactly how long the virus can last, but probably in an outdoor environment, maybe a day at most. And so I think it's obviously the health experts are telling us that washing your hands frequently, trying to be cautious of making sure you wash your hands before you touch your face after you've interacted with a surface that could be contaminated with the virus, That's all excellent advice. As far as how long to do it or how long to let it sit, I think most people feel like the data around food containers and furniture and things like that are that the virus probably can't last more than a day, something like that on that. So if you wanted to, that's why I think you can use your best judgment of how soon you're going to be sitting in that same table or how soon you're going to be touching that same container of food to help guide your actions there. But washing your hands, wiping down things certainly are great public health measures to help protect you and other people that come behind you.
And if I can just add to that, we were setting up a biosafety lab to handle SARS-CoV-2 virus. We had to go through very extensive contamination validation procedures. We had to develop these procedures to make sure that the waste that we were generating did not contain any live virus. And one of the things that we've observed, and also this is the same recommendation from the CDC, is that the virus is very rapidly inactivated by a whole host of disinfectants, including alcohol, including bleach-containing substances, substances also containing acids. And so what we found is that the common household disinfectants that are available are really highly effective and can neutralize the virus in just minutes. And so again, the place to go look for that detailed information is on the CDC website. They have recommendations there for what kind of disinfectants to use and also how long they should be applied to a particular surface. It's just great information. But we kind of felt that the virus was fairly wimpy towards disinfectants, and that anything that we tried, it was very effective at neutralizing the virus pretty quickly. So
excellent. So our next question, I think a lot of people are concerned about their likelihood of catching coronavirus, but they're wondering what possibly they could do to bolster their immune system. What drugs can a 70-year-old take for COVID as a precaution to have in your system before possibly getting COVID? For example, hydrochloroquine, quercetin, zinc, and the like.
You want me to take this one?
Sure.
Okay, so as a 70-year-old myself, maybe I should actually take this one. Basically, I don't think there are any real recommendations about medicines to take to prevent SARS-CoV-2 infection. If you take supplements such as vitamin D or antioxidants, then I would continue to take these. But I think you've got to be careful about adding more drugs or supplements to your daily regime, because drugs and even supplements can have side effects, and they could be of different severity depending on the person that's taking them. I would encourage people to talk about changing their routine on these things with their healthcare provider before taking any new drugs or supplements. But I mean, I would reiterate what I think Dr. Moore said earlier, that following the precautions that we know about, the use of masks, social distancing, cleanliness, washing, etc., are probably just as important, in fact, more important at the moment to do that sort of thing.
On to our next question. The president was treated with monoclonal antibodies. This seemingly was an important therapeutic addition. How available is this to everyday Americans? Dr. Sexton, do you?
Sure, yeah. So these, the monoclonal, President Trump received several different things. He received remdesivir, which is an infusion-only drug that has to be delivered by intravenous injection. And so, again, received these monoclonal antibody therapies. And so these are not currently FDA-approved drugs, so they're just not available to the public, which is unfortunate. But the nice thing that Dr. Loring mentioned is that there is a tremendous amount of late-stage clinical trials that are happening to establish both safety and efficacy in human beings. And so he received 2 monoclonal antibody therapies, one, both by Regeneron. One was an ACE2 monoclonal antibody, and the other one was a spike protein monoclonal antibody. So one is a human protein, is a viral protein, and that cocktail together has shown promise in clinical trials. And but they're not available yet to the public, and but after the phase 3 clinical trials and with all of the effort that the FDA is putting into scrutiny to make sure that anything that they release is going to be safe and effective, and they're doing it very, very quickly. I think there's a good chance to have a very efficacious monoclonal antibody therapy. I mean, those are generally very expensive. We've got lots of different kinds of drugs from, as John mentioned, we have dietary supplements, we have small molecule drugs, and then there's biologics and vaccines. And so we really need all of these things. And monoclonal antibody therapies take a lot of expertise to develop, but they're well on their way. And so I think it's a very promising modality for therapeutic intervention for SARS-CoV-2. So it seemed to work for the president. So
great. I think we have time for one more question. How long will it take to develop a safe and effective vaccine? And how long between the time of the vaccine development and widespread distribution in the US? Dr. Loring, I know you've been thinking about this a lot.
Yeah, so no one knows. I think, I suspect we already have 3, 4, 5 vaccines that are in phase 3 trials. And these are the big trials that look at safety and how well a vaccine works. And so that's, these have 30,000 people. So these are really big trials going on, and they're already well underway. I suspect we'll have some, data from those by early in the new year, for sure. And, they'll probably come in at different times. And then we'll know, at least over a short term, how well they work. And then we'll probably start seeing some of them get the go-ahead, so some form of approval for use. And then it'll probably take another, 6 months more, I would imagine, before they're widely available. There's a lot that goes on in terms of how you produce that much vaccine and how you distribute it. And part of Warp Speed has been to go ahead and spend money on producing these things before we know if they work. And so it's part of the goal to get them going. And so, but it's still, there's a lot of logistics, and some vaccines are harder to make than others. So I think there's a big unknown there, but I would say within a year.
Great. Very encouraging. So thank you to our panelists for offering your valuable time and expertise. I want to thank all of our attendees for your engagement and interest. We really hope you found this session informative and that you've learned some things that might be applicable to your lives. I want to extend my sincere thanks to our wonderful donor community and our foundation and corporate partners, and a special thanks to those of you who have joined us throughout the summer to learn about Michigan Medicine's response to the COVID-19 pandemic and other important issues. We look forward to keeping you well aware of our activities in the months to come. As a reminder, a recording of the webinar will be sent in a follow-up email, and we will send a list of helpful resources as well. Please join us for our webinar on November 12th at 1 p.m., in which our experts will discuss diabetes care and research in the era of COVID. Please stay healthy and safe this fall. Thank you, and go Blue.